protein biochemists, we often refer to mutated proteins or mutant proteins, but mutations actually happen at the level of DNA, not at the level of a protein. And this makes a big difference. This really matters in particular fields. But as protein biochemists, we do so much work with different versions of proteins that we need simple terminology in order to describe them and refer to them. And so we typically just talk about mutant proteins as compared to wild type or kind of the normal version of proteins. And we do this because we have so many different versions that we're studying. And typically we're making those versions to study to figure out how proteins work. So I want to introduce you to some of the notation that we use. So I'll go into more detail. But basically for a substitution where you're changing one amino acid for another amino acid, you are going to give the location of the change. And before that, you're going to put what it was originally, and then after the number, you'll put what it was changed to. So G91A would indicate that at the 91st position of the amino acid sequence, you went from a glycine to an alanine. So you substituted a glycine for an alanine. So this is what's there now, and this is what was there. For a deletion, what you're going to do is you're going to use this delta sign to indicate the amino acids that were deleted. And so say if amino acids 80 to 100 were removed, we'd write this as delta 80 to 100. So the delta indicating the change, and the change is that we've removed the amino acids 80 to 100. And we make lots of different versions of proteins to study for different reasons, and then we can refer to those different versions as constructs. And they can be really helpful for figuring out what different parts of proteins do, as well as for stabilizing proteins in order to kind of get a picture of them. Not really a picture, but do techniques like X-ray crystallography, where we need the proteins to be really steady and sturdy and things. So let's go first start though by why there is this distinction and why this distinction does matter, that the mutations are happening at the DNA level and not the protein level. Not all mutations in genes are actually going to alter the sequence of the proteins that they encode. And so there are multiple reasons for this. One is that they might be in non-coding intron or other regulatory regions, and another is that the mutations might actually be synonymous. So really quick, let me go through what I mean by this. We have to use this term coding versus non-coding when we talk about DNA. Coding means that it has instructions for putting amino acids in the correct order to make a protein, and non-coding indicates that it's a region that does not contain the instructions for actually sticking those amino acids in order, but it contains some sort of regulatory information maybe. And so there's regulatory information at the ends of genes, as well as interspersed between exons, which are coding regions that are actually included in the mature messenger RNA that's made. So you have a gene, it's got these exons and these introns, and then the introns get cut out and you get your final mature messenger RNA after some processing. What can happen is you can have mutations in those introns, and because those don't have the actual instructions for placing together amino acids in order, then they're not going to change the sequence of the amino acid if you were to just make a little change, usually. What can happen, however, is that those can actually influence how those exons get spliced together. And so there are splicing mutations that can cause these exons to get skipped or include introns or partially skip exons. And so this can be a big problem with some diseases such as spinal muscular atrophy. And so lots of things like that. And it can also introduce frame shift mutations where you really skew the whole reading frame. And so you end up with a lot of amino acids in the wrong order. And so when we talk about mutations at the genetic level, we can refer to things like synonymous and non-synonymous, as well as frame shift mutations. And so we're talking about changes that we have in the DNA that may or may not alter the protein that they encode. What happens is that in the process of making a protein, the ribosome, so the protein making machinery, knows what sequence to put things in because it's following the instructions in the messenger RNA. The instructions are coming in the form of these codons, these three-letter words, and then transfer RNAs that match those words, they're complementary to those words, are going to come with the corresponding amino acid, and the ribosome is going to put that into the chain. You can see that there are multiple different ways to kind of spell some of these amino acids. There are multiple codons for the same amino acid. And so we say that the code is degenerate. And what this means, therefore, is that there are some changes that you can make to the sequence of the DNA that aren't actually going to impact the sequence of the protein. They could impact how fast the protein was made or things like that, but they're not going to alter the actual sequence of the resultant protein. We call such mutations synonymous. And so here we can use the term mutation because we're actually talking at the level of the DNA. And we use this term synonymous to refer to a mutation that isn't changing the amino acid sequence. This would correspond to no change in our protein. And there are changes, however, that are non-synonymous. And so the non-synonymous is where you're changing one codon to a codon that means a different thing. And this could be a missense mutation. And a missense mutation is where you're going to swap one amino acid for another. And so that would correspond to a substitution in your resultant protein. For example, a non-synonymous mutation in the gene for beta hemoglobin causes a substitution in the resultant protein that changes a charged amino acid that really likes to be on the surface to a non-polar amino acid that is typically going to be excluded from the surface of the protein, and this can then therefore cause the proteins to clump up and cause problems. You also have what we call a nonsense mutation, and a nonsense mutation, you're changing the codon for one amino acid for the codon that says stop. And so some of the codons are stop codons, and that tells the ribosome to stop making the protein. And if you make a change to the DNA that then makes a change to the RNA copy that's made, then that could introduce a stop codon where you don't want one, and then you're going to get a short or truncated protein. For a nonsense mutation, you would often see this indicated with an X. And so instead of E6V, which was saying that you were changing a glutamate to a valine at the 6th position, you would have something like E6X, which was indicating that you had a stop codon in place of that glutamate at that 6th position. You can also have indels, insertions, and deletions. If you insert or delete a duplicate of 3, then your protein's going to either be missing a letter or have extra letters, but you're not, then on either side it would be okay. Whereas a frame shift mutation, you make a change that gets things out of frame, and so it messes up that kind of 3-3-3, and therefore your whole sequence after that is going to be different. Now let's get back to thinking about things at the protein level. And so we often make a lot of different versions of proteins. And so we can use things like introduce mutations to DNA that then will cause the changes in the proteins that naturally happen in different disease states. Or we can make changes that are going to then allow us to test what different parts of a protein do, or that are going to make the protein behave more nicely when we're trying to do techniques like crystallography. The normal or unaltered version of the protein we refer to as the wild type, and then we refer to the altered proteins typically as mutants, even though they're not technically mutated, it's the DNA that's mutated. The protein is just altered. Often what we actually use is we use this term constructs. And so a construct is basically a version of a protein or a version of the genetic instructions for the protein. So maybe a version of the wild type sequence for the protein, but with an extra part on the ends that's going to serve as a purification tag, such as a his tag or a strep tag. And we might generate lots and lots of different constructs, especially if you're in a field like structural biology, where you're trying to explore the interplay between the biomolecular form and function. We use techniques like site-directed mutagenesis to make changes to the sequence of the DNA for the protein in order to make changes to the resultant protein. So we do a lot of site-directed mutagenesis to try to figure out what different regions of proteins do, as well as make proteins that are going to be more amenable for some experimental techniques that we do. Thanks to recombinant DNA technology, we can make these specific changes to that DNA. We stick that DNA into bacteria or into other cells, tell those cells to make the protein for us, and then the protein is going to incorporate the changes that we instructed the cells to do by mutating the DNA in that specific way. and then seeing what happens to the resultant protein. And so we might be doing this to kind of certify the protein for crystallography to try to get to crystallize. Or we might be doing things like making just a certain portion of the protein, like a domain, in order to study it in isolation. We might be adding one of those tags, or we might be trying to introduce mutations in the DNA that will then mimic mutations that happen in different disease states and things like this. So lots of different reasons why we might want to make these mutations to the DNA to make all of these different quote unquote mutant proteins and proteins that not necessarily mutated. And remember, the protein itself is never mutated. But even if the sequence of the protein that you're studying isn't altered, you might actually have extra regions on the end that are going to serve as those purification tags. And so for lots of different reasons, we might refer to a construct. We use that term construct to refer to both the protein as well as the DNA that is encoding for it. So like the plasmid or whatever, like we could refer to this as a construct or the resultant protein. So we use this term kind of loosely, just like we use the term mutant pretty loosely. But remember that the mutation is actually happens at the DNA level, and it may or may not cause a change to the protein. And this distinction, although it might seem pedantic, it actually is important in particular contexts, especially when you're thinking about clinical things, you're thinking about evolution, all these various things. Not all mutations are actually going to cause changes in proteins, even if they're happening in a region of a protein coding gene. And even if they're happening in a coding region of a protein coding gene. And so that technicality is important, but for practicality, with protein biochemists, we often talk about mutated proteins. So let's get back to talking a little about some of the types of changes. So substitutions, again, we put the original letter first and what it's changed to afterwards. So a change of the 91st amino acid from a glycine to an alanine would be written as G91A. Or that mutation in the sickle cell anemia, where we have that mutation in the beta globin gene, it's happening at the 6th amino acid position in the sequence, and a glutamate is being replaced by a valine. So we go from a glutamate, E, to a valine, V. So this is why it's really important to know those one-letter amino acid abbreviations, especially if you're a protein biochemist. And so that's one of the most crucial things to kind of memorize is those abbreviations. Insertions, that's where you add some letters. And so these are kind of harder to specify in the shorthand. But insertion mutations can happen and cause diseases, and such as Huntington's disease. We do, however, deal more with deletions. And so deletions we can indicate with that delta sign. So if we remove amino acids 80 to 100, we'd write delta 80 to 100. Sometimes what we're doing is we're shortening the ends of proteins, and so we often refer to those as truncations. So if you cut off the starting end, we call it an N-terminal truncation. And if you cut off part of the ending end, we call it a C-terminal truncation because we talk about proteins as going from the N-terminus to the C-terminus. And note that I'm using terminus, so the ends are called termini, or each is called a terminus, not a terminal. And so you can have an N-terminal region and a C-terminal region, and you can have an N-terminal truncation or a C-terminal truncation, but those are happening at the N-terminus or the C-terminus, with the terminus if they're happening at the very, very end. And we might be doing something like that in order to cut off those floppy regions, which are typically at the ends, and can make it so that the proteins won't cooperate when we try to get them to freeze in place for X-ray crystallography or things like that. But remember that the actual changes are happening because of changes that are made at the level of the DNA. So the mutations happen in the DNA. They may or may not cause changes in the protein, but when we're introducing the mutations, we're typically doing it so that we make those changes in the protein. Although sometimes we do make mutations in the DNA to just make it so that the DNA is optimized for different cells to make it for us.